Hi.
Hello, can you hear me, Professor Kim?
Yes, I can hear you very well.
Thank you very much. Thank you very much for accepting our invite for
Oh, you're
this
welcome.
TPCF webinar. And before I actually start introducing you to the audience, I'm just going to briefly talk about TPCF. So Tehran University of Medical Sciences Preclinical Core Facility. And we are sort of a core facility where we have various different imaging modalities, and we are interested in bringing the scientists together in a multidisciplinary platform where they can perform experiments for their projects. And on top of that, we are sort of having various different educational programs such as symposiums, webinars, and various different workshops. And it's an honor to have you as probably, I think you're the, I don't know, I think sixth or seventh presenter. And we are having these TPCF webinars once every month. And with that, I would like to introduce our tonight's guest, Professor Jean Kim. And Professor Jean Kim is actually a professor of biomedical engineering and radiology at Weill Cornell Medical College at NYU in the United States. And Jean's interests are focused on three areas: development of dynamic contrast enhanced MRI methods for assessment of tumor treatment response, as well as mapping the extracellular space using diffusion, time dependency of MRI experiments in cancer and normal tissues. And his third research interest is role of saturated fat in breast cancer development and progression using MR spectroscopic imaging methods. With that, I would like to give the platform to Professor Kim. And thank you very, very much for accepting our invite, and really looking forward to hear your interesting talk.
Thank you so much for the kind introduction. It's my pleasure to share some of my research with all of you today. Good evening. I know the time is quite late over there, but hopefully I can keep you engaged with this topic that I'm so much excited about. This will be diffusion MRI today. As just introduced, I've been working on the different part imaging modalities for the cancer imaging, but today we'll be focusing on the diffusion imaging. I hope that everyone can see my screen.
Yes, we can see it. Perfect, thank you.
All right, great, thank you. So the diffusion MRI probably has been known to many people who are working in the brain imaging, especially with the name diffusion tensor imaging, which I will not be talking about too much today, but I just want to briefly to share with you. For some of you who are not familiar with diffusion imaging, diffusion tensor imaging has been really the game changer in the neuroscience and brain studies by providing this fantastic imagery that reveals something about the brain structure that we have not been able to see before without diffusion imaging. And obviously, as many of you know, that this itchy box cell, by using diffusion MRI, we are mapping the pattern of water diffusions, and particularly in the brain structure where there is a fibrous structure, you will see the major direction of a water diffusivity into one direction. And that has been encoded, using color or within different shapes. And this is from my old study in 2005. So you've probably seen more impressive images than this one before. But nevertheless, it's a similar information content. And these kind of diffusion tensor imaging has been successfully used to study how our brain has been wired in a sophisticated and very complex ways. But without using any destructive technology, you could actually look at the brain structures. But today, what I'm going to focus on is the cancer imaging, which is not as colorful and impressive as this kind of diffusion tensor imaging in the brain structure. But nevertheless, it has been quite useful and helpful imaging modality when it comes to cancer imaging. This is the one example in the head and neck cancer squamous cell carcinoma. This is ADC, apparent diffusion coefficient map, is showing the tumor structure, intrastructure, which is quite different than what you could see in the other conventional imaging modalities. And particularly this low diffusion area is known as solid tumor with this good contrast. So it could be very helpful to understand how the tumor is made of in terms of a solid tumor versus a necrorigocystic part of tumors. And this technique has been used to also look at the treatment response to the chemotherapy or radiation therapy. So we conducted one study quite some time ago with about 80 patients who are going through the chemo radiation therapy. This is the result that I'm showing in terms of ADC apparent diffusion coefficient. We did a scan before therapy and 1 week into therapy and at the end of therapy. And we looked at their treatment response and grouped them into 2 groups. One group with a complete response without any evidence of a disease, residual disease, and the rest of them as partial responders. When you group them in this way, we were quite surprised to see and also happy to see that the separation between this complete responder and partial responder, even before therapy, you could probably tell that their differences, and this could be sort of indicating that diffusion can be used as a predictor of the response to a certain therapy. And also, you can notice that there is a significant increase of diffusivity within even 1 week of a therapy, especially for this group with complete responders, whereas the partial responders didn't show any changes in this short time of 1 week. So maybe this early change could be another thing that could be very useful to predict how the patient will do at the end of the day. And there are multiple other studies by other groups showing similar results, but maybe in a different scale of diffusivities, similar settings. But the question that most of us have with this kind of exciting result is that what is really happening in the tissue? And many people have a different way of explaining this ADC changes, but most of people think in a way that the diffusivity inside of a cell is probably not very high. And diffusion measurements we are doing is probably dominated by diffusivity in the extracellular space. So the extracellular space increase by cell shrinkage could lead to maybe ADC increase. Whereas if the cell swells and extracellular space reduced, and maybe you will see the ADC decrease. It's kind of simplistic scenario, but could be explain many of the cases. But wouldn't it be nice if we can actually measure the cell size or extracellular space size, and also the diffusivity inside of cells and outside of cells separately, so that we don't have to speculate, but we can actually measure these kind of specific biomarkers directly from the tumor cells. So that's the first question of the talk. Is it possible to measure cell size and extracellular space volume non-invasively? And this is one of the 2 questions that we'll be focusing on today. So we'll talk about maybe this topic for the next 20 or 30 minutes or so. And of course, the answer in my mind is yes. And I like to convince you by introducing this method called the POMACE, which has been developed by my previous postdoc Olivier a couple of years ago. So we'll talk about something a little bit more about what this long name really stands for. So let me step back and think about what do we need to do when we design diffusion MRI experiment to measure the cell size. And probably the first thing that we need to do is to think about what's the characteristic diffusion time, the diffusion time that matters to the cellular structure. And you can think of that as through some simple thought experiment. So let's think about this simple cell structure, kind of round cells. And if you have a water molecules with a short diffusion time and how much it will diffuse through to the long diffusion times. And you can quite easily think this kind of pattern of the displacement length or mean square displacement. So when the diffusion time is short, your distance that water molecule can travel will depending on the diffusion time quite linearly. Whereas if the diffusion time keep increases, most of the water molecule will experience the restriction by the cell membranes. So it will be limited by certain length, certain mean square displacement, which reflects the cell size. So there's 2 components in this curve, right? The later part is the cell size dependent components, But the initial part is a diffusivity dependent component. You probably need both of them to really figure out how the cell's size is, because we are not measuring the displacement directly. We are measuring the diffusivity in the MRI experiments, which depends on the length as well as the time scales. So if the cell size is about 10 micron, you can think, and diffusivity is about 1 micrometer square per milliseconds. Your characteristic diffusion time could be about 100 milliseconds. It's a kind of just run thumb calculation. So that means that you need some the diffusion experiment before this time as well as after this time to capture the deflection going through this characteristic diffusion time in order to measure the cell size more kind of in a meaningful way. Of course, this is a much bigger than the tumor cells. Tumor cell will be probably in the order of maybe 5 micrometer in terms of radius, or maybe smaller. And there, if you do the same simple calculation, you probably have about 25 milliseconds as your characteristic diffusion time. And it could be even shorter than that, because the diffusivity could be actually much higher than 1. I'm just using 1 as a simple way of calculating to get a just quick sense of how big how the time should be. And when it comes to neuronal cells or the axons, which is about 1 micrometer maybe diameter, not even the radius, it could be a much shorter time range that you will have to have in order to have that pattern, that a characteristic pattern that we need to measure the cell size. Okay, so from this thought experiment, what you could sort of agree with me is that we need a range of diffusion time around the characteristic diffusion times. And for the cancer cells, probably 10 milliseconds might be the time that we will need. And diffusion times longer than 10 milliseconds, you can usually use the conventional method called the PGSE, pulsed gradient spin echo sequence. But shorter than that, we need some special technique called the OGSE, which I will talk about a little bit more in the next slide. The pulse sequence, the commonly used for diffusion MRI experiment, including DTI, is based on the pulsed gradient spin echo method, which uses a pair of diffusion weighting gradients. In this case, the diffusion time will be defined as the time between these 2 pulses. And there are many technical challenges of making this short in a clinical scanner. Mostly because of the gradient amplifier, gradient strength is not strong enough. In animal scanner, which is about 10 times stronger gradients than the clinical scanners, you could probably go down to a few milliseconds, but not much more than that. So we need some special technique called the oscillating gradient per sequence, which is sort of which is in this way that you have to use oscillating gradient, as the name reflects. By using this one, you can go down to close to about 1 millisecond. Of course, the downside is that we cannot make the B value or diffusion weighting strong enough, because there's a factor that have to be considered when it comes to these oscillating gradients. Okay, so without going into too much technical detail, let me show you one of the animal experiments that we did. So we set out to do the experiment using these techniques. We used the GL261, the brain tumor models in mouse, 7 Tesla scanner, Bruker scanner with a 750 millitesla per meter. So once again, if you have a Prisma scanner with 80 millitesla per meter, it will be about 10 times stronger. If you have a older scanner or the latest one, it will be about 20 times stronger than the clinical scanners. Just give you a sense of the difference between animal scanner and the clinical scanners. And we have multiple diffusion directions, at least 3 directions to average out, and also different diffusion times and oscillating frequencies. So here is one, 2 examples of the data for 2 different diffusion times. This is for the PGSE, the pulse gradient spin echo sequence data, where we have more room to play out the stronger diffusion weighting. And also this is the OGSE data. the one thing that we noticed is that there was no clear IVIM or perfusion effect. For some of you are familiar with IVIM. We didn't really see in this model, so we could just use a whole range to estimate the diffusivity from this data. So basically, the slope of this curve will be the diffusivity that we can measure from this particular diffusion time. For OGSE, also we looked at the different directions. This is actually showing all three direction measurements, X, Y, Z directions. The data sort of overlaps very well between to each other, so showing that no preferential directions in terms of diffusivity when it comes to tumor cells. So from this kind of pattern of measurements, we are measuring one diffusivity per diffusion time. If you combine those together, my apologies that this one shows as in terms of a frequency, 1 over square root of a frequency, but you could sort of think of this as a time scale on the x-axis and y-axis as the diffusivity. And this is the brain of a mouse and the tumor on the top. And these black dots are showing the diffusivity changes as a function of diffusion time. So it kind of shows this kind of pattern of diffusivity changes that as we expected. And this open circle is from the gray matter area on the away from the cancer tumor the tissue. And as you can expect, that in the range of diffusion time that we are doing experiment, we don't see this deflection of the pattern, diffusivity patterns. You will have to go down much further, much shorter than 1 milliseconds actually, to see these patterns. There are actually few studies that people have done it to actually measure the neuronal cell size at the very, very short diffusion times, but not in our study, we can see that. Okay, so the question is, what do we do with this data set? How do we extract information of a cell size and extracellular space, volume, fraction, things like that from these data sets? One thing that I like to do is to sort of separate this curve into two regions. One is this earlier time points where the things seems to be quite linear in terms of a square root of diffusion time. So for this short diffusion time, once again, you can think of it as a simple cell structure where most of the water molecules that starts from the middle of the water, the cells will diffuse around, but they will never feel or they will never experience any restrictions. Whereas some water molecules near the cell membrane will probably likely to experience some restrictions. So these two sort of scenarios can be used to measure certain structural properties about these cells. And this has been used in the porous media physics for quite some time already. And they use exactly this concept to make the model based on the two pools of water molecules. And you could sort of drive an equation that describes the diffusivity of this whole system in terms of these two compartments, which involves the surface to volume ratio as well as the free diffusivity. And these two measurements, the parameters can be estimated from if you have a multiple sort of diffusion time measurements that sort of gives you these two water pools. Okay, so we apply this model to this time range in the short time diffusion range. For oscillating gradient sequence, you have to use certain the correction factors, but this can be derived from the sequence property itself. So it's not necessarily a tissue property other than the dimension D. And these are the maps that you could actually generate just to use only the short diffusion time. You could generate the free diffusivity without any restriction. It's becomes about close to 2 in most of the tumor cases. Surface to volume ratio. So this is again 1 over sort of radius of the cells, so to speak. So this will give you some sort of a range of the size of structure that restrict the water diffusion in these cases. So I like to probably one thing that I like to point out is that this experiment was the first experiment to show this short time linear dependence to the square root of diffusion time in vivo tissue. So before this one, this technique has been used for maybe porous media in other areas, but not for the in vivo tissue. So we are quite excited about seeing this kind of pattern in vivo tissues. In order to sort of confirm whether what we see in the in vivo is really the correct reflection of substrate volume ratio as well as the D0, we performed the ex vivo experiment using the special coil called the histocoil. So this is a specially made coil, very thin kind of a plate with a slit that can, the histology slide can just slide in this way. So this is actually like just a light microscopy image of the one section of the tumor in the brain. And this is the MRI experiment of the same section that you could have matching images showing the tumor in here as well. And as you can see in here, we see the similar pattern of diffusivity changes in the same time scale. And this blue one is a control data in the PBS, the saline outside of a sample that it doesn't show any diffusion time dependency at all. Once again, this intercept, it will be D0 that you can measure, and the slope is reflecting the substrate volume ratio that we are measuring. So combine this in vivo and ex vivo data together, what can we learn? We're looking at the D0, the free diffusivity that we measured from this experiment in vivo, ex vivo room temperature, ex vivo at 37 degrees. As you would expect, room temperature, the D0 will be decreased. And then when you bring the temperature to 37 degree again, it will go up. And that can kind of match with the in vivo case as well. So I think this pattern sort of confirms that D0, the measurements of the free diffusivity measurements make sense. What about a substrate volume ratio? So there's some quite striking substantial difference between the in vivo and ex vivo cases in terms of the substrate volume ratio. I think that's probably due to the changes when you do the sectioning. Also, since we have to hydrate the sample again, there could be some expansion of a tissue within the slide, which probably ends up with smaller SOV or larger cell size in this case in ex vivo scans. But one thing that you can notice that when you change the temperature to 37 degree, SOV or cell size measure doesn't really change significantly, especially the green one, which use the shorter diffusion time, and the white one with a little bit wider range of diffusion time. The shorter diffusion time range one shows more consistent measurements. So this kind of supports that the SOV measurements as well as the D0 measurements using this short diffusion time kind of makes sense. And obviously, if this is a measurement of just an intracellular space alone, then we could use this one to estimate the size of the cells. But unfortunately, when you do this kind of experiment with a whole tissue, we have a mix of intracellular as well as extracellular space. And in the same thing, this short time behavior of diffusion experiment, diffusion will happen in the extracellular space as well. So we cannot really convert this one to radius, but we need to do something more than that. So let's go back to the data that we have so far. So we used only a short time regime to extract the two parameters. What can we learn from the rest of the data that we have? And the rest of the data with the longer diffusion time, now that we can assume that most of the cells, most of the water molecules in the cells will experience some sort of a restriction or full restriction by the cell membranes. And there will be extracellular space water molecules that are going through the space between the cells, but they are not fully restricted. They are just hindered. They will have maybe reduced the diffusivity in overall together. So we probably have to consider these two components together when we try to interpret the data outside of the short time regime. And that's exactly what we did. We look at this combination of these two components in terms of signal that we are measuring. But we can also approximate that the diffusivity that we estimate from the total signal will be also some of the diffusivity of intracellular space as well as the extracellular space, assuming that the V value is small and we are only operating this experiment in the the regime that doesn't involve the higher order of the B values. Among these components, the intracellular diffusivity, since we know that if you assume the spherical shape of the cells, we could actually predict how diffusivity will change in terms of diffusion time using the diffusion equation known as a Fick's law. And also you are using the boundary condition for the spherical shapes. You could actually come up with the analytical solution for diffusivity for the oscillating gradient, as well as the pulse gradient pulse sequence. And in this hairy looking complex equation, what you could sort of notice is that there is a size information R that is the parameter that goes into this equation, as well as the intracellular water diffusivity. And the other ones are all sort of pulse sequence parameters that comes into it. Okay, so using this equation, you can sort of calculate how the diffusivity will change when the frequency decreases or diffusion time increases toward the left-hand side. It will go to complete to zero if you just keep increase your diffusion time, because the length is fixed. It will come to completely zero in terms of the apparent diffusivity of the intracellular space. So that's the case for the intracellular space. But when you look at the actual data, and if you pay attention to the y-axis, the decrease of the diffusivity that we are seeing is not all the way to zero yet. It's only up to maybe about 30% decreases around this time range that we used. So suddenly we haven't gone all the way to here. Maybe we might be somewhere in between here. But there is the extracellular component that's probably affecting also quite significantly, which doesn't go all the way to zero as well. So we could assume that there's extracellular components that sort of changes, and we know that there's other part of physics that I'm not going to discuss today, but it can give you some prediction of how much of extracellular space diffusivity will go down depending on the structure, the type of structure that we have. And this never goes to zero in many cases. So that the change in the extracellular space will be relatively much smaller than the intracellular space diffusivities. So what we did was that we used the intracellular diffusivity using this sort of analytical solution that we have. But in terms of extracellular space, we assume that this will be constant, even though this is probably there will be some change within certain range before it goes to infinite diffusion time. For within this short diffusion time, relatively smaller range of diffusion time that we have, we assume the extracellular diffusivity will be close to constant. That's what we have. So this comes to the model that we have. And we have these four unknowns: the cell size, intracellular diffusivity, and extracellular space volume fraction, and extracellular diffusivities. Okay, so we did some simple simulation study to see whether we could measure these four parameters when you have this kind of diffusion data. Once again, this is just displaying the data, same data in a slightly different way by displaying the frequency, not the 1 over a frequency, but this time it's just a frequency. So if you just convert this one to a time scale, this will become again the same pattern that you see. So my apology for using just the plot that we have, which is not consistent between the different slices. But anyway, so we assume certain parameters of tissue structures and added a noise to the data and try to fit this range of data that outside of short diffusion time to our model that we have, and to try to estimate these four parameters. And this is what we have. Somehow, with even a small amount of the error added to the data, we couldn't really measure the ground truth, which is a red line. It all goes to the sort of boundary conditions. So to solve this problem, one way that we come up with is to use the short time data, the OGSA data, which provides information about the free diffusivity. And free diffusivity will be again, sort of the sum of intra and extracellular diffusivities with volume fraction as the weighting factors. So we constrain this estimation process to using these conditions. And by doing so, we were able to measure this one quite accurately, given data, sort of error statistics matching to the in vivo data cases. Okay, so that's in a nutshell the method called the pulsed and oscillating gradient MRI, because we need both techniques for assessment of cell size and extracellular space measurement, or in short, POMACE. So this is a case of 3 different tumors. We need a short diffusion time to estimate the D0, and a long diffusion time, relatively long, but still quite short, about 31 milliseconds only up to. That will give you a measurement of extracellular space, as well as the cell size measurement in vivo. So this is all in vivo measurements that I'm showing. Also, we did extra ex vivo measurement as well to match. Okay, so that was quite exciting that we were able to do this. And by doing in vivo ex vivo measurements, we saw quite similar range of cell size measurements. Once again, extracellular space seems to be expanded in the ex vivo measurements. We tried to validate this method using electron microscope. Although, I mean, it's quite challenging to find the exact location that match with MRI, and also the field of view is so much different, so you cannot do much, but we try to just match as much as possible. And what we come up with is the cell size is about around the 5 micrometer in the EM images, which also matches with in vivo, ex vivo MRI, the data. So we are quite convinced that we should be able to measure the cell size using MRI experiment, as well as the extracellular space, which is also a very important component. We are currently trying to assess this technique, although it's not completely yet, in the treatment response monitoring studies. In this case, we are doing the 2 MRIs, one before, another one after the therapy, using bevacizumab or Avastin, as well as the 5-FU. In this pilot study that we've been doing so far, we found that the extracellular space decreases after this one cycle of therapy quite significantly, whereas in cell size it seems to be increasing. So this is one scenario that we are seeing with this particular therapy method. Then hopefully that we can apply this method to other types of therapy to see how the cell size and extracellular space changes. So this is an ongoing study, and hopefully I can bring more results later on. But also I was quite impressed by other group at the Vanderbilt. They are also doing something similar diffusion experiment, but they've been doing also more extensively, much more so than what we have been doing. But what they have demonstrated recently in this study, which I think will be show up in the MRM in a few months, they used this 3 diffusion times with multiple V values, which shows in this plot. And using this set of this is kind of sort of a reduced set of experiment than what we did, what I showed you in the previous slides. Using this limited data, they were able to map the cell size in the breast cancer images, as well as the intracellular volume fraction, extracellular diffusion coefficient. Based on their study, the intracellular volume fraction as well as the extracellular diffusion coefficient have a large variability, but at least the cell size seems to be quite precise. So they were quite excited about using this technique, bring this technique to the clinical settings. I think that's very important, and also very encouraging as well. Okay, so that was kind of a very brief introduction of how we can use MRI techniques, especially diffusion MRI technique, to measure cell size and extracellular space volume non-invasively. And I hope that I convinced you that the answer is yes, it is possible. It is possible with animal studies, but it is also possible with the clinical scanners with some limitations. It would be great if we could sort of validate how the measures that we get from the MRI technique using different modalities. The validation and also bring it to the clinic will be the big task that we need to do more in these cases. I'm going to switch gear to the second question that I have, but also I'd like to say that if you have any question, please feel free to interrupt me, unmute and interrupt me, because I cannot see the chat at the moment, but I'll be more than happy to answer any questions if you have any.
Thank you very much, Professor. I think it's better to leave the question right towards the end, and
okay.
The audience will gather all questions and we'll ask them towards the end. I think that would be best.
Makes sense. Okay, all right.
Thanks,
all right, so I will spend, okay, thank you. So I will spend maybe next another 20 minutes or so for the next question, which is how about the water exchange between a cellular and extracellular space? And you may notice that I was assuming the cell membrane is an impermeable membrane, which is not the case. We know that the cell membrane is permeable to water molecules and other ions content as well. But we had to use this assumption in order to solve this problem in those experiments. And you may notice that we used our longest diffusion time used is about 30 milliseconds. So the exchange effect will be sort of minimum or negligible in those cases. But of course, we know that exchange happens throughout the whole time scales. But can we actually measure them? If we go to longer diffusion time, this could be a confounding factor, but can we actually maybe rather measure this water exchange rates? And what could that mean if we can measure it? So let me go back to the clinical study that we had earlier with the head and neck cancer study that I showed you in the very beginning. And at that time, what we also had was the dynamic contrast enhanced MRI, where we could use the tau I, intracellular water lifetime. So this will reflect how long water molecule stays in the cells. Also, it will give you a sense of how fast it exchanges between intracellular and extracellular space. By using this technique, also we measured pre 1 week and also post therapy time points. And at that time, we didn't really see any significant difference between the complete responders and partial responders in terms of a tau I or water exchange time rates. But when we look at this data about 10 years later, when the most of patients have about 8 to 10 years of a follow-up, we look at their overall survival in terms of all these MRI parameters that we measured, especially before therapy. And it was quite interesting to see that the pre-treatment tau I was the one that the most predictive of long-term survival in our data, at least within our data set. And as you can see that the patient with longer diffusion, the tau I, which means a slow diffusivity, they did very well in terms of long-term survival compared to those patients with a short tau I or fast water exchange rates. So what does this water exchange really means? So the water exchange, once again, that I'm talking about is water exchange through the cell membranes, and there are many different types of water exchange measurements that concerns the vascular, through the vascular, the walls, or maybe different kind of structures. But within cancer, I'm kind of ignoring that the vascular space will be very small. Intracellular, the cellular membrane, the exchange through the cellular membrane will be the main components that we will be able to measure. And this water, the water exchange rate, or one of water lifetime in the intracellular space, will sort of have two main factors. One is the membrane permeability, and the other one is the cell size, once again, surface to volume ratio. So if you can measure so this water exchange rate could be also be affected by cell size change as well. It may not be just a permeability. So what are we really measuring in actual tissues? So even though we cannot really answer this question directly, there is a very nice review paper by Charlie Springer a couple of years ago. He looked at different studies and look at the cell size changes in those studies compared to the water exchange rate increases or changes with the same challenges. And as you can see in this plot, in these 4 example of studies, the cell size changes is only about maybe 10 to 20%. I guess that makes sense. Like mechanically, you cannot really expand suddenly by 100%. Whereas in water exchange could actually change maybe 100% or maybe even more than that in many cases. So the water exchange could be maybe a more sensitive measure of whatever change it might be happening in the cells. That's one message. One study that I really like about among these studies, the study by this Zhang et al. Using the yeast cells in the perfused NMR tube. So he was able to measure tau I or 1 over tau I, which is the exchange rate, while you are perfusing the cells using oxygen. And also you're measuring the volume fraction of cells. Or also in this case, using the MRI and NMR techniques. And they changed oxygen to nitrogen. So effectively they are shutting down the ATP productions. And so you can confirm the ATP was depleted by using this phosphorus NMR techniques. At that time, it's quite interesting that the water exchange decreased by maybe about half, about 50%, whereas the volume fraction maybe increased by around 20% or so. So that you can see the water exchange is more sensitive, has a bigger effect that you could measure. But also you can notice that the water exchange doesn't go all the way down to 0 when you completely shut down the energy productions. And of course, water exchange doesn't have to depend on the energy in the cells. It could happen through aquaporin, the protein in the cell membrane, without using any energy. So we know that that's happening. And that seems to be accounting for about 50% of water exchange. But that experiment that I just showed you sort of indicate that there is other significant portion, maybe another 50% of water exchange happening using the energy in the cells. And there's many ion, sort of energy dependent the ion channels that also co-transport the water molecules. And as far as I understand, we don't fully understand this water transport between the inside and outside of cells using the energy in the cells. But this water change could be an indication of cell metabolic status. So maybe linking, sort of speculating between this and to the study that I showed you in terms of long-term survival, maybe our patient who had a slow water exchange before therapy, maybe they had a less aggressive tumor, they responded much better. But whereas a fast exchange, maybe they had a lot of energy, very active water, the cell metabolism happening even before therapy, ended up to the poor survivors. Okay, so that kind of like sort of my rationale and the reason why I'm so excited about the water exchange and why I wanted to measure water exchange also using diffusion DCE MRI method, but also using a diffusion method, because DCE you have to inject a contrast agent. Also, there are some technical challenges when it comes to measuring the water exchange rate using DCE technique that I showed you in the other slide. But today, since it's a diffusion talk, I'd like to share some our recent result of measuring the water exchange rate using diffusion method. And this technique is based on the diffusion kurtosis measurements. And just give you a brief sort of a background about the kurtosis measurements. So this diffusion measurements in terms of p-value, diffusion weighting have sort of an exponential decay that you can measure the diffusion coefficient. It's called the Gaussian regime or diffusion tensor regime. That's sort of corresponding to the first order Taylor expansion or cumulant expansion of actual signal behavior. And the full signal description will require high order terms that will be required to measure or explain the diffusion behavior at the higher diffusion weighting. And by doing so, you come to the term called the diffusional kurtosis, the K term, and K will tell you what's the sort of diffusion sort of distribution pattern in the tissue. It's not really the Gaussian all the time. If K is higher than the 0 or positive value, it means it could be a super Gaussian, or it could be maybe a sub-Gaussian depending on the certain tissues, although it may not be quite easy to measure in the in vivo cases. Okay, so nevertheless, the K is something that we are interested in for the following reasons. So K shows the diffusion sort of patterns in the tissue, but what does it mean when it comes to the multiple compartment? Let's say if you have an intracellular compartment, extracellular compartment. And there has been some study to show what this K means in terms of the multi-compartment diffusivities. If there's no exchange once again, the kurtosis is actually the variation of diffusivity between the different among the different compartments. If it's a two compartment, the kurtosis also corresponds to the difference between the two diffusivity of two compartments. What if there is an exchange, just like the tumor cells? If there is an exchange between tumors, the two compartments, diffusivity will become constant at some point. If you have a long diffusion time to allow any water molecules in one compartment have a same sort of the probability to cross the membrane and go to the other compartment. It's a long way of saying that long diffusion time. Then your diffusivity becomes constant, and your kurtosis will decrease as you increase the diffusion time at the rate of diffusion, the exchange rate or exchange time. So diffusion, the kurtosis changes can be a way to measure the exchange time based on if your diffusion times are long enough. Okay, so I know this is not a simple concept, but let me just leave it there and show you some actual data. So this is the experiment that we did. I think it's probably also the first data that we showed in the in vivo tissue that we can use the long diffusion time to go to the cargo region where the diffusivity becomes completely flat or constant. So if you recall, a few slides earlier, I was showing the diffusivity changes in terms of diffusion time at the short diffusion time ranges. But we are now moving to the long diffusion time where diffusivity becomes constant. And this is the regime that that I just described, that the cargo model is valid. And in this regime, the kurtosis changes monotonically. And this can be used to measure the exchange time, or it can be converted to exchange rate as well. But exchange time will be the term that I will be using for the rest of the talks. Okay, so we sort of repeated this experiment with the multiple animals as well as the different tumor models. I'm just showing you one tumor model types. In all cases, it's quite noisy, of course, in an actual real world experiment. But you can appreciate that the pattern, consistent pattern of diffusion sort of becomes a constant in the long diffusion time, and kurtosis becomes sort of monotonically decreasing. So when you combine together, you could actually have a better trend, and also you can measure the exchange time individually as well as the group wise. We also have demonstrated this technique in the breast cancer patients. This is the invasive ductal carcinoma in both cases. And you can see the diffusion, the signal, there's enough signal still at the long diffusion time with a quite strong diffusion weighting. You could see the same pattern of constant diffusivity and kurtosis changes, which allows us to measure kurtosis as well, the diffusion exchange time as well. And going forward, what we are interested in at the moment is applying this technique to the head and neck cancer patients who are going through the chemo radiation therapy once again. And we like to use this technique to see whether we can predict their long-term response, which what I demonstrated using the DCE experiment earlier. And this is one example of diffusion data. I just want to show you the quality of data as well. This takes about 15 minutes to get all these images. At the end of a study, probably we don't need all these diffusion times. We probably need maybe some of them, but we are using all these images at this point as an early stage. Once again, if you look at the data, the blue one is showing the muscle cell data in terms of diffusivity as a kurtosis. Since the muscle fibers are much bigger than the tumor tissue, you see the decrease of the diffusivity within this diffusion time that we are using, and an increase of kurtosis as well. But in the tumor tissue, once again, the diffusivity is almost flat, so confirming that the diffusion behavior is in the cargo regime, which allows to apply this kurtosis equation to kurtosis decrease patterns. And this we can measure the exchange time in this case. Also, exchange time of about 100 milliseconds in the tumor, I think is what is expected based on the previous studies. All right, so just last few slides is just to give you a quick result what we have from our clinical study at this point. We are running this study with HPV, human papillomavirus positive or P16 positive oropharyngeal squamous cell carcinoma patients. These particular cohort of patients are known to be a good responder, very sensitive, but a good responder to the radiation therapy, and more than 90% of patients usually have a good the outcomes. So the question is, can we de-escalate the radiation dose for those patients? So we've been doing this experiment, this study by looking at their response in the middle of the treatment and reducing their therapies. So we are monitoring their diffusivity changes in this course. Most of patient case, just briefly, you can see that the red one, which is the de-escalated patient groups, have lower median diffusivity compared to the control cases who didn't really show a good response in this cohort. But most significant was in the kurtosis at the week 4 time point. Most of patients with a good response at a higher kurtosis than the control patients. What most interesting to me is the exchange time measurements. And we haven't really seen significant differences between the de-escalated patient versus a non-de-escalated patient. All of them supposed to show a relatively good response. But among this patient, there's one particular patient who kind of failed within 3 months after the completion of treatment. And the rest of them are doing quite well so far. And this one patient who failed, we noticed that before we de-escalate, which is a week 4 time point, the exchange time was very short compared to the rest of the groups. Once again, the short diffusion time means a fast water exchange. There's many theories of recent studies showing that there's a repopulation of cancer cells as a resistant cohort of cancer cells growing quite aggressively at a certain point of therapy. We don't know for sure whether that's the case for this case, but once again, it looks like we are seeing some patients who are failing later on have a short exchange time or the fast exchange during therapy as well. Okay, so that's I think one of the interesting applications of diffusion imaging that we could use going forward. Just as a summary today, we looked at the 2 questions. One is whether we can measure cell size or extracellular space volume, as well as in the second question, we looked at the possibility of measuring the cytolemmal or cellular extracellular water exchange rates. I think the answer that I have, I think the yes for both, and hopefully I convinced some of you that that's the case. But big question that we have is, of course, how do we validate that we are measuring translation is always another big question that I showed you a few cases in our study as well as the Vanderbilt study. We're trying to bring this technique to the clinical studies. We could always cut little bit corners or optimize a certain way to make it clinically feasible sort of a technique. But whether it's clinically or preclinical studies, how do you validate these measures? I think this is a big question that I have. And hopefully some of you who are working with different imaging modalities in a preclinical settings, if you have some good question about how we could sort of validate some of these measures, please let me know. I think that will be a very exciting part of a study that we can do maybe in the future. Okay, with that, I'd like to thank many people within my group who are in my group before as well as outside of groups, but wanna especially thanks everyone in my group. Today I focused on the diffusion imaging study that focused on cellularity, but also our group is specialized in the vascular measurements using DCE and other techniques, as well as looking at the stromal effect on the tumor, especially in the breast cancer using hormonal effect as well. And I'm just showing you some of the key members in our team that are sort of working on this area, especially thanks to Olivier and Maren, who worked on the part of the study that I presented today. So with that, I'd like to stop here and take any questions if you have.
Thank you very much, Professor Kim. Actually, I really enjoyed your talk, and I know we are all sort of kind of separated because of this COVID-19, and this is a virtual session, but I'll have a round of applause and I ask everyone to do it. So probably they share that and send the energy to you. So I've got a few comments, and very, very interesting that you showed very beautifully pure physics and mathematics actually come in hand to actually help us to understand more about the biology, especially about sort of, we thought, okay, cells are very important, intracellular signaling molecules are very important. Now we're coming to realize, okay, extracellular compartments and also the diffusion rates are very important as well and can give us very interesting clues. So I really enjoyed the sort of the marriage of biology, physics, and the mathematics in your talk, and that was very interesting and stimulating for me. I've got some questions. One was regarding, so with regards to, you mentioned it briefly as well, when we have a heterogeneity of in terms of cells, what will happen in terms of how can we get a consistent sort of result? Because there's so many various different cell types. We have immune cells, we have various different blood type cells, and having them together and creating a sort of a very multifactorial and variations, how can we sort of deduct a conclusion out of it?
Absolutely. I think that's an excellent question, and question that also really mind boggles me for quite some time as well. Because we know that in a cancer cell, it's not just a pure cancer cells. There's a huge amount of macrophages, as you just mentioned. There's the M1, M2 populations, and there's vascular cells. So yes, it is a gross approximation that we are doing. This will be, at the moment, this is just average of our cells that we are measuring. We do get some population sort of distribution information out of these measurements. But we don't have the specifics to certain cell types yet. So I think that's the excellent question, and hope as also subject of a future study. How can we optimize and fine tune this technique to really tune into certain population of the cells? I think that will be a very interesting study going forward. So sorry that I don't have an answer, but I completely agree with you. That should be the future direction of the study.
Okay, cool. We have got another question from Neda Zakir, and she's asking, is a cell size and extracellular space can differentiate between different kinds of cancers? So she needs, probably I think she has 2 points. One, is it different between different cancer cells, which you showed in various human setup as well, if that was the case. And another question maybe I can add to that, I think this is what she's asking, is once we have cancer stem cells differentiating, does that differ as well, do you think?
So in terms of difference between the different cell types, different cancer cell types, there have been some cell studies or sort of a study based on the surgical samples. They're measuring the actual cell size and things like that. They are showing some differences between the different cell types, but not a huge, as you could imagine. There were also overlapping quite a lot. So I think people who have been working on this measuring the cell size, especially with the in vivo method, not so much focusing on the differentiating the different types of tumors at this point, but since this might change more dramatically during the therapy, I think this might be a better technique to look at the therapy-induced changes, where we have a baseline to begin with, so that we don't have to wonder where it started from. I'm sorry, I didn't really quite get your second question. Was it about the differentiation of the cells?
Cancer stem cells, yes.
Cancer cell differentiations.
Yeah.
Yeah.
Exactly.
So
probably the same answer.
Yeah, probably same answer. One thing that the sort of a next level or one idea we've been playing around a little bit, but we haven't been able to do the study more thoroughly yet, is can we measure more subcellular structures like nuclei size or mitochondria size, things like that. If you have a subcellular size information, I think this might lead to some answer close to grading or maybe a differentiation of cells, or more relevant to those kind of questions. So I think still is unexplored area. I think will be also quite challenging. I haven't thought too much about that, but excellent area to think about more. Yeah.
There's a question by someone called BRZ. I don't know, it's an abbreviation. So he or she has thanked you for your presentation and asking to see if you've mentioned various different advanced mathematical modeling, and he or she is asking if you know any interesting books for advanced mathematical modeling. If you could actually mention some of them, would be helpful for this
Right. Oh, absolutely. I think if you are not familiar with the diffusion MRI, or even if you are familiar with diffusion MRI, probably this diffusion MRI book edited by Derek Johnson. I think this one has a good combination of some mathematical background as well as some clinical sort of application as well. There's a good chapters by Kiselev, Valery Kiselev, as well as the Peter Callahan. So Callahan, he has also his book, which is also good at the sort of the microscopy method as well. So I think those two books probably the best one, Callahan and Derek Jones, I should find out.
Yeah, cool. I've got some other questions, and whoever from the audience, if they're interested, they can raise their hand up, and the admin would give them access to the microphone, or also they can type the questions. I've got some of the questions myself. So because you're interested in sort of the water molecules outside the cells, so when we have a sort of let's say I'm talking about a mouse or a rat experiment we're doing, when the animals are sort of dehydrated, do you expect to see different in signals as well, or difference in parameters? And so do we need to have a new setup and a model for that?
Wow, okay, that's excellent. A very interesting question. At least we know during sleep in our brain, extracellular space increases by 10 to 20%, right? That's something, the new finding that everybody got so excited in the brain study. So if there's such a significant change of extracellular space, I think that the diffusion experiment should be able to pick up that. Within brain, it will be slightly different conditions that we have to think about compared to the tumor, but yes, it should be possible. I'm not sure whether dehydration will lead to that much of a change. I mean, that seems quite dramatic change. So you will really have to dehydrate quite a lot to have that much of an extracellular space water content change. But I'm not quite sure how much actually leads to the extracellular space changes by dehydration.
Cool.
So it will depending on that.
Mm-hmm. Yeah. I've got another sort of maybe question or comment that I was wondering if you actually looked into it. So you mentioned about permeability and cell size. I think this could be a very interesting point, especially for performing various different kinds of tumor imaging. or even in terms of therapeutics, where we inject sort of nanoparticles, where they, based on the sort of high permeability, these nanoparticles, like therapeutic nanoparticles, would go inside the tumor, and then with an extracellular, extra as in external wavelength, or I don't know, pulse, then we can actually activate these nanoparticles and have a sort of a heated nanoparticle somehow to get rid of the tumor. So maybe this is a very interesting sort of imaging that you mentioned, and a very good calculation, I would say, that we would perform first to see how permeable the tumor is. So in brackets, we could have a personalized medicine in a sense as well. Then we would inject those nanoparticles, and then we would actually look for imaging the tumor, or even sort of having a tumor therapeutic effect, anti-tumor effect based on those nanoparticles.
Absolutely.
Have you looked into this as well or
not? I haven't studied anything specifically based on the certain sort of type of therapy yet, but immunotherapy is the one thing that I have mind, and we kind of in the stage of setting it up, where once again it's not a exogenous particle, but endogenously we know that there will be the change of macrophages, and that could be something that we could measure. But also, I mean, you bring a very interesting point that when we actually inject the different types of nanoparticles, how does that change the cell size or morphology? That could probably also be detected by this kind of technique as well. So I think we probably haven't, I mean, this technique is just barely introduced to the community at this point. So we haven't really explored all the possible potential area that we can use. Of course, at the moment, I mean, even the theory and all the results are very promising. There are a long way to go to make it as a quite accurate, precise tool for all this measurement in vivo. I hope that we get there sometime soon.
Well, I hope the same as well for you. And with that, I would like to end the session. And thank you once again, Professor Kim. It was an honor having you and listening to interesting, very, very interesting talk. And looking forward to have you in our sort of symposium that we have, our annual symposiums. So this year would be our 4th annual symposium. And we would like to hear sort of more about your interesting research if you're interested in our symposium.
Absolutely.
And
that would be great.
Yeah, thank you very much. And with
You're
that,
welcome,
I would like to thank the audience, which was more than 100 individuals. Professor Kim, you were looking at your presentation. I was looking at the numbers. It was plus 105 individuals. And with that, I would like to once again thank Professor Kim for his time and for his great research and sharing it with us. And also thank all the participants who actually stayed with us. And I would like to ask the admin if they could actually present the next speaker as well. So everyone would realize the date and the topic. And with that, I would end the session. And yes, so we have preclinical imaging of brown adipose tissue and pancreatic islets by Professor Reza Mivoluki, and he's at Southern California University of Health Sciences at the United States. Thank you very much, everyone, for making this event very interesting, and special thanks to Professor Kim. Thank you very much, Professor.
Thank you. Thank you, everyone.
Thank you. Bye-bye.
Bye.